Okay, question one, extractions and washings. So first we're going to talk about extractions. So liquid-liquid extractions can be used to purify compounds and separate their mixtures. So this would involve exchanging certain compounds between two liquid phases. So one would be the organic phase and the other one would be the aqueous phase. These are most likely immiscible in each other, meaning that they don't dissolve in each other. And so one layer would sit on top of the other. And so the organic layer would be on top and the aqueous layer would be on the bottom. And the one on the bottom would be the more dense layer. So typically we would be attempting this to separate an organic acid or base from a neutral compound, and this would be done using a solvent. We're trying to get a compound of interest out of one solvent into the other. So right now would be getting it into the organic solvent. So the first step, we have them sitting on top of each other, one sitting on top of the other with the compound of interest in one of them. And we would be using a separatory funnel normally when we're doing this. So here we're going to have them mixing together. And then after they settle, you can kind of see that the solvent has kind of extracted into the organic solvent after we've mixed them together. And so most of it is present in the organic solvent now. And lastly, we can see when we separate the two, we can see that the compound of interest that was once in the blue solvent is now present in the pink solvent. And you can kind of see that there's some left in the blue solvent. But in order to further purify it and to get the rest of the compound of interest out, we would kind of perform the whole extraction step again until we have all of the compound of interest present in the organic solvent, which is the pink one. So in washing, you're going to want to remove a specific compound from a mixture. So this is going to be, again, using a solvent, and what you're going to be removing can be maybe a contaminant or whatever. So what you're trying to get rid of should be more soluble in the solvent than in the mixture. So for example, say you're washing clothes. The dirt is going to be more soluble in the water than the clothes are, right? So the dirt is going to go away, and what you're going to be left with are the clothes only. So here we're going to have our two phases here. So right now we have in the blue phase, we have contaminant here, which our contaminant is going to be the black dots here. So in the separatory funnel, like we did before, when you mix them together, when you shake it, you're going to have, you're going to wash your aqueous layer here, and the contaminants are going to be moved into the organic layer. And so you're going to be left with these two layers, and when you separate them, you can see that the contaminants is now in one solution, and the compound of interest that you wanted to keep is still in the other solution. So now if you wanted, you could discard of the solution with the contaminants and you're left with what you wanted. Both of these techniques require a separatory funnel and two immiscible compounds, but the main difference between them is when you're washing, you're kind of purifying something by getting rid of the contaminants, and then you can get rid of the phase that contains those contaminants and then keep the other one. And then with extraction, you are taking your compound of interest from one phase and you're transferring it to the other using a solvent. Question 2, unknown mixture 2. Piperidine and oxalic acid are soluble in water while anthracene isn't. So we're going to add water to the crude material. So we're going to add water to all three of them. And since oxalic acid and piperidine are soluble in water, we're going to be left with a solid of anthracene. So we're going to observe an aqueous solution of piperidine and oxalic acid form while a solid of anthracene is left over. And so we can also use a vacuum filtration if needed to separate those two compounds. So basically, a vacuum filtration would use a filter paper and a vacuum suction, and it would vacuum the water and the aqueous mixture of the water, oxalic acid, and piperidine down through the filter paper because they're liquid, so they're small enough to go through the pores of the paper, and they'll leave the solid anthracene sitting on top of the paper. The anthracene can then be dissolved in acetone, and we're going to use the simple distillation method to purify it because the solvent and the solute both have a boiling point difference of over 100 degrees Celsius. So we're going to have the acetone and the anthracene dissolved in each other, and the one with the lower boiling point is going to heat up first and it's going to evaporate first. So that's going to be the acetone. So it's going to evaporate and it's going to go through the condenser and it's going to end up here. And the anthracene, which has a very high boiling point, is going to stay here because it's not hot enough for it to boil away yet. So now we're going to work on the separation of the other two compounds. So this technique can also be used for the other two compounds that are right now in an aqueous mixture. So piperidine has a lower boiling point, so when distilling, it's going to evaporate first into a gas and it's going to be condensed and it's going to return back into this area right here. So we're going to have the piperidine, oxalic acid, and we remember that's all dissolved in water right now. So the water and the piperidine both have a lower boiling point than oxalic acid, so they're going to both evaporate, go through the condenser, and they're going to end up here. And again, the oxalic acid, which has a way higher boiling point, is going to remain at the starting. Now we have all three of the compounds separated. And since the water and the piperidine both have similar boiling points, they might have been evaporated together, so further purification of the piperidine might be necessary. And we can do this by using a drying agent such as saturated sodium chloride, which is brine that'll crudely remove the rest of the water. And then after that, we're going to add a drying agent such as anhydrous sodium sulfate, which will remove the rest of the water, further purifying the piperidine. Question 3, Organic Synthesis Lab. So for the procedure, we used a distillation flask with sodium hydroxide, 2-naphthol, 95% ethanol, and also magnetic stir bead to ensure everything was mixed properly while it was boiling. We then added a condenser and we boiled everything until it was all liquid. After that, we added 1-iodobutane, and when that was all done, we poured the mixture over ice to bring it below its boiling point so we could wait for the solid to start forming. We then chose to use a vacuum filtration method to isolate the solid component. So vacuum filtration, basically, you're filtering the liquid through the filter paper using a vacuum, and everything that is too big, so the solid particles that are too big to go through the filter paper, stay on top, and so we separate the liquid and solid. And so at the end of this, we did a TLC, which is a way to tell us how far the reaction had progressed and whether or not the starting material was consumed. So the TLC results showed 2 spots for the final product and 1 spot for, marking one low spot that was marking the initial reactant, which was the 2-naphthol. So this means that a new product was formed throughout the reaction, and the ending product was thought to be butoxynaphthalene because first we made naphthoxide ion, which was our strong nucleophile, by deprotonating the 2-naphthol and the sodium hydroxide. And then we added the electrophile, which was the 1-iodobutane, which gave us an SN2 reaction to take place and formed butoxynaphthalene. The IR spectrum showed strong CO bonds, so carbon-oxygen bonds, which were around the frequency of 1000 to 1300, and also medium carbon-carbon alkene bonds, which were around the frequency of 1600. And so these are the 2 main bonds that are found in butoxynaphthalene, and this further confirms the identity of the final product. The reaction involved a nucleophile, naphthoxide, attacking the electrophilic carbon of the alkyl halide, iodobutane, displacing the leaving group. This SN2 mechanism required a nucleophile that was good enough to attack the carbon, and our nucleophile was naphthoxide, which was an ion as well as being a very strong nucleophile needed for an SN2 mechanism to work. The substrate was also not sterically bulky. In fact, it was a primary alkyl halide, so it was readily accessible to the carbon center for easy substitution, and also so that the nucleophile could attack the carbon center as a leaving group dissociated. Question 4. The first technique I'll be talking about is extraction. So, extraction refers to the transference of compounds from a solid or a liquid into a different solvent or phase, and this is used as a step to purify compounds. Drying. This is usually done following an extraction or washing, as there's usually water left over, therefore, the compound is not pure. First, the organic layer is washed with brine to crudely remove the rest of the water before the drying agent is added. And so, when the drying agent is added, if it moves around freely, that means it has dried the substance, but if it remains clumpy, more drying agent needs to be added for further drying to occur. The rotary evaporator. At the end of a reaction, a solvent might have to be removed, so the rotary evaporator is one of the most efficient ways of doing so. So the rotovap has three parts: a cooling source, a vacuum source, and the rotary appendage. The reaction vessel with the compounds is rotated and heated in a hot water bath, while the vacuum reduces the vapor pressure and so that the solvent is going to boil at a lower temperature. And as the solvent leaves, it goes through a condenser which cools it down, and so that we can have the solvent collected elsewhere. And this is another purification method. TLC. This is a chromatography technique used to separate non-volatile mixtures. So it's done on a sheet of plastic with silica gel thinly spread over it, and this is called the stationary phase. So a spot of the solution is going to be put down at the end of the plate, and then it's going to get developed in a jar of the mobile phase, which is called the eluent. The eluent will then slowly move up by capillary action, carrying the spots that were initially put at the bottom. This can be used to monitor the progress of a reaction. It can be used to tell us more about the purity of something as well as the relative polarities of those substances. So if the spot is lower on the plate, that means it's more polar because the stationary phase is polar and so it was attracted to it. And on the other hand, if the spot is higher up, that means it's less polar because the mobile phase is not very polar and so the substance was attracted to that. Gas chromatography is another chromatography method used to separate and analyze compounds. So it's commonly used to test the purity of a substance or to separate the different compounds of a mixture. This also has a stationary and a mobile phase where the compounds stick to the stationary phase because of dipole interactions, ionic interactions, as well as dispersion forces. Organic compounds flowing out of the chromatography column are seen as peaks on the chromatogram. The amount of time this takes for the substance to leave the column is also calculated, and this can help to deduce the identity of the substance. Recrystallization is a purification technique for solid compounds. So where the solid would be first dissolved in a solvent and then it's going to be later precipitated out. The principle behind this is that the amount of solute that can be dissolved in a solvent increases with temperature. And the solute has to be insoluble at room temperature but very soluble at a higher temperature, so when it's boiled or heated in a hot bath. A solution is made by dissolving a solute in a solvent near its boiling point, and insoluble impurities are then going to be filtered out, and the solution is then cooled. So when the solution is cooled, what's going to happen is that the solution is going to be saturated at a much lower concentration of solute than before. So whatever can't stay in the solvent is going to form purified crystals. And this solution can then be filtered out and you'll be left with a purified compound. So the melting point can help to tell the purity of a substance. A melting point apparatus is used with small amounts of a substance that is melted in a controlled way. And so the experimental melting point can be compared to the literature melting point. And if the experimental melting point range is too broad or lower than the literature value, then either the sample is contaminated or the heating was too rapid. So a simple distillation is a technique where two liquids of different boiling points of over 100 degrees can be separated. So the solution is heated, and then the substance with the lower boiling point is going to be evaporated first. And then the vapor will go through a condenser and the liquid will be collected elsewhere. So for example, here we have water and oxalic acid. So the water has a lower boiling point, so it's gonna evaporate, go through the condenser, and then it's gonna be collected here, and that is simple distillation. IR spectra analysis. So the bonds in atoms constantly vibrate and rotate by absorption of energy, and this energy can be found in the infrared region of the electromagnetic spectrum. So these bonds all have their own absorption frequencies, and the IR spectrometer will measure the amount of light that is transmitted through the sample. So for NMR, the sample is dissolved in a solvent, and the chemical shifts of the sample are dependent on the solvent that is chosen. So when molecules are placed in a strong magnetic field, the nuclei of some of those atoms are going to behave like small magnets. And if a broad spectrum of radio wave frequencies are applied to the sample, the nuclei will begin to resonate at their own specific frequencies. And these frequencies are measured and converted into an NMR spectrum, and the frequencies will be shown as peaks. And so NMR can be used to deduce the content and purity of a sample as well as its molecular structure. And this can be done by using IR spectra analysis as well.